Hey Team Bio, so today's screencast is about what happens when enzyme structure goes wrong, when proteins denature. Okay, more on what that means in just a second. Okay, so you're all comfortable. You've seen before this model for enzyme action. It's called the induced fit model, and we all drew it in class. It starts out with an enzyme. This enzyme has a three-dimensional shape. It's folded in a really specific way to get this shape. This looks like a tertiary structure protein because it's not composed of multiple subunits. There's only one polypeptide chain folded up here. But let's say this is catalase. The enzyme that you know breaks down H2O2 and forms H2O and O2. That's the unbalanced chemical equation of the reaction that catalase catalyzes. Okay, so if this is catalase, then this substrate must model hydrogen peroxide. So you know what happens in the induced fit model. The substrate binds the active site of the enzyme. The enzyme undergoes a conformational change. Which is just a fancy way of saying it undergoes a shape change. So it changes its shape, and in changing its shape, it catalyzes the reaction. So in this case, it breaks the bond that's shown here. Okay, so enzyme undergoes a conformational change, and then the reaction is catalyzed. And then the enzyme releases the product. And it reverts to its original shape. And this is important because it's ready to catalyze the reaction again, as long as there's more substrate. Okay, so what happens when this breaks down? Let's chat about it. Okay, so here we have our model of catalase. For whatever reason, we've changed the color, so these are representing the same enzyme here. But now we can really see the folded structure of the protein. I said it's a tertiary structure protein, so it's only composed of one subunit, but you can see here that we've got some motifs that you know. So here is a beta sheet, this region right here. So let's put it in a box. Nope, try to put it in a box. Nope. There we go. So this region of the protein is our beta sheet motif. And we've got a couple different alpha helices. I'm just going to circle or put one in the box. So here is an alpha helix. And these little dotted lines that I'm showing indicate hydrogen bonds. So this is a hydrogen bond, and these are hydrogen bonds. Okay, you all remember from our exploration of water that hydrogen bonds occur between two different molecules or two different parts of one molecule. When we have an area that has a slightly positive charge, and usually That slightly positive charge is hydrogen, but it can also be nitrogen. And that slightly positive charge interacts with a slightly negative charge somewhere else. And we call this interaction a hydrogen bond. This is a relatively weak bond. And it can break. If it's disrupted. There are a couple different ways to break hydrogen bonds. First of all, when you, what do I want to start with? Okay. The first thing that can disrupt hydrogen bonding is the pH of a solution. So the pH of a solution, if it is below 7, we call that an acid. The pH is measuring the concentration of hydrogen in a solution. So an acid has more hydrogen ions than normal floating around in the solution. So we'll say H plus has increased. Now, I'm sure you can imagine if we have a situation where we have a lot of free hydrogen and we have bonds formed between areas of a molecule that have a positive charge or a negative charge or a slightly positive charge or a slightly negative charge, these free hydrogens in solution could diffuse and interrupt this bond by forming their own bond with the slightly negative region of the protein. So when there's a lot of free hydrogen in solution, then these hydrogen ions sneak in here, they get in the way of this hydrogen bonding with this oxygen, and they form a hydrogen bond with this oxygen instead. Okay, so when the pH is low, when it's an acid, it means hydrogen concentration is actually very high, and this bond can be disrupted. When the pH concentration is high, this means that we have a base. And instead of having a lot of free hydrogen in solution, now we have a lot of OH minus in solution. More than normal. So the same thing happens, but in the opposite direction. If we have a bunch of free OH minus, it's going to diffuse through the solution. It's going to get in the way of this bond, but instead of bonding with the negative oxygen, it's going to bond with the positive hydrogen side, but it still can break this bond. Okay, so having a solution that's too acidic or too basic can break a hydrogen bond. Another way to break a hydrogen bond is to have a bunch of ions. So, for example, Na plus or Cl minus. And the same kind of thing happens in this situation. When you have a lot of free sodium ions, they can diffuse through the solution, they can form an interaction with this negatively charged oxygen, and then this bond will break. When you have a lot of free chlorine ions or chloride ions in solution, the same thing's true. They can diffuse through here, they can interact with the hydrogen, and then they break this bond. Okay, the last thing that can disrupt hydrogen bonding is the temperature. When the temperature gets higher, when it gets hotter, there's just more energy in this mixture. The temperature is a measure of the average movement of particles in solution. And when the temperature is hot, these atoms are moving around a lot faster, and hydrogen bonds break. So, hot temperatures can break hydrogen bonds. Cold temperatures don't break hydrogen bonds. They just freeze hydrogen bonds in place. So, cold is not going to destroy the H bonds. It's not going to break them. It's just going to lock them into place. You all know that in order for enzymes to work, they have to undergo a conformational change. They have to change their shape. And so as an enzyme gets colder and colder and colder, it can be harder and harder to catalyze its reaction because its shape gets really rigid and it can't undergo this conformational change as easily. So when an enzyme is frozen solid, generally it won't work because it can't change its shape. Okay, but these three other situations, when a protein is in an acid, when it's in a base, when it's in a solution with a bunch of ions, or when it's too hot and H bonds are broken or disrupted, this is what we call protein denaturation or proteins denature. Okay, and what does that actually mean? You know that the primary structure of this enzyme, all these amino acids are linked together by a covalent bond called a peptide bond, and it's very strong. But the shape, the secondary and tertiary shape of this enzyme is held together primarily by hydrogen bonds, and they're pretty weak. When the protein encounters an acid, base, lots of ions, or hot temperatures, these hydrogen bonds will break. So the protein denatures H bonds break. And we go from a folded functional protein to an unfolded non-functional polypeptide. So here, all of the hydrogen bonds have broken, and so the shape of this enzyme has been lost. It's now just a long, straight chain of amino acids, and it's no longer folded in its specific shape to have its active site fit the substrate. So now we're left with a broken protein. Can't do its job anymore, can't catalyze its reaction. And this process is irreversible. So once a protein denatures, it can no longer go back to being a folded functional protein. And the reason for that has to do with how they're created. More on that in the spring term. But for now, I want you to know that when a protein denatures, this is an irreversible shape change. It's lost its shape, can no longer catalyze its reaction, and it's done, toast. When a protein freezes, these hydrogen bonds are locked into place, but if you warm it back up again without getting it too hot, you can rescue a frozen protein, and it can work just the same as it did before if you warm it back up gently. Okay, that's all.